Thank you to introduce my presentation. I'm Marianne QUEMENEUR of the Mediterranean Institute of Oceanography at Marseille. I'll talk about the microbial diversity of hyperalkaline springs of New Caledonia producing hydrogen and methane. In such environment, both hydrogen and methane are produced naturally by an abiotic process called serpentinization and other associated reactions like Sabatier or Fischer-Tropsch. And both molecules can be used as carbon source and energy source for microorganisms living in the subsurface and in deep environment. This type of environment associated with the serpentinization are very interesting because they make it possible to address a fundamental question such as the origin or the limit of life on Earth and other planets. And also, they make possible to address applied questions such as the search of extremophile for biotechnology use, and also for the question of CO2 storage, and more recently for the search of natural hydrogen production, also called white hydrogen. Now, this type of environment can be found in different places around the world, mainly in Europe and also in Asia, also in North America, and also along the Mid-Atlantic ridge, where the most famous site, Lost City, a submarine site, was discovered in 2000 at 800 meter below sea water. And this submarine site produced high concentration of hydrogen and methane, and also hyperalkaline fluid. And it is very different from the black smoker that produced acidic fluids. And this fluid forms carbonate chimneys where very specific microorganisms live. And we have also a similar site, a similar submarine site. We can find this site in the New Caledonia in the South Pacific. And this site is located in the Prony Bay, New Caledonia. We can find similar chimneys of calcium carbonate. And also this site produced a high concentration of hydrogen, methane, and anoxic fluid. But this site is different from the Lost City site by the low temperature of the fluid, and also by this location at low depth, less than 50 meters below seawater, and the release of low salinity fluids. For more than 10 years now, we study this hydrothermal field, and we collect samples in intertidal area and also in a submarine area. All these sites release hyperalkaline fluid and variable concentration of hydrogen and methane, and variable concentration of these gases depending on time. And we also extend our research to on-land spring in New Caledonia. In different sites, we found a different concentration of these gases, but the highest concentration of hydrogen and methane were found in the Prony Bay in New Caledonia. In the frame of a project Microprony, our objective was to describe the microbial diversity in this site and to isolate new microorganisms with biotechnological interests. Our objective was also to determine the role of microorganisms in the cycle of hydrogen and methane. And we also investigate the microbial group involved in the formation of the chimneys to understand the emergence of life on Earth. To do this, we collect fluids and chimneys. From this sample, the DNA was extracted, then sequenced to identify the microorganisms. The microbe was also quantified by PCR or microscopy, and we perform enrichment and isolation of microorganisms using alkaline and anaerobic media in tubes and also in bioreactor. And we monitor the growth and the consumption of hydrogen and methane, and the production of this molecule too. Inside these chimneys, we can find many microorganisms and a low diversity of archaea dominated by uncultivated methanosarcinales and dominated by a specific phylotype of this order, only found in this type of environment. This phylotype has not yet been cultivated, and we think they potentially produce methane using hydrogen or oxidize methane in anaerobic condition. But we isolate a new species of hydrogenotrophic methanogen. It's a new genus new species of the genus Methanobacterium, and this species is adapted to alkaline pH and frequently found in this type of environment. We found diverse bacteria in this environment, dominated by hydrogen oxidizing bacteria. In oxic condition, we found aerobic hydrogen oxidizing Hydrogenophaga, and in anoxic condition, we found sulfate-reducing bacteria from the Desulfonatronum genus. And next to this hydrogen oxidizing bacteria, we found also bacteria of the phylum Firmicutes and closely affiliated to Clostridia, potentially able to produce hydrogen biologically in an environment that produces already hydrogen. We isolate different species in this environment, able to ferment sugar, organic acid, amino acid, and one of them is able to produce hydrogen in high quantities, notably the species, the strain ProH2. In conclusion, in this type of environment, we found the presence of hydrogen oxidizing microorganisms, a diverse population, and the presence of firmicutes, potentially able to produce hydrogen. We found a change in microbial community depending on environmental factor. And we have a different question that we develop in the different projects, in the project Microprony, but also in the Hydromic project. It's how does hydrogen shape the community, and what are the main acceptors of electrons used to oxidize hydrogen in subsurface, and what is the importance of Clostridial in the hydrogen concentration measure at the surface. We also continued the research in New Caledonia, and we developed a drilling program to analyze the microbial community, but also the different molecules of gas in the subsurface. Thank you for your attention.